Bismillah ar-Rahman ar-Rahim. This is a description for the data show slides of the bone histology. This presentation actually it comprises the slides given in the year 2014 and 2015 for better training of our students. In this presentation, we will see the two types of the bone, which are the compact bone and the spongy bone. You should remember the sites for each type, and you should also remember the different ways to prepare the sections of the bone. Here we should also know that it is formed of organic part, and this organic part, the organic part here, organic part constitutes 35% of the structure of the bone, and it is formed of type 1 collagen, in addition to ground substance formed of two things, glycosaminoglycans and glycoprotein. So in the ground substance we have two materials, and in addition to the collagen type 1. As regards the inorganic, we have the salts of the calcium, mainly calcium phosphate and carbonate. Okay, they are present on the surface of the collagen fibers. Here. This is the collagen fibers, so these are present on the surface of the collagen fibers, as well as in the ground substance, as we have said before that we have a ground substance also formed of glycosaminoglycans and glycoprotein. This inorganic substance constitute 65% of the bone structure. And the calcium salts are responsible for the hardness of the bony matrix. If you look to this picture here, these are the calcified tissue. This is a canal called Haversian canal, and these are empty spaces that are called the lacunae, in which we have the mature bone cell, which is called the osteocyte. And this is the arrangement of concentric lamellae of the calcified collagen fibers, alternating with the rows of the lacunae containing the osteocytes. And all these circles have one center, so they are called concentric lamellae. This picture is a light microscope picture for a transverse section in decalcified compact bone. Why it is decalcified? As it is stained. In this photo, you can diagnose this skeletal muscle, transversely cut skeletal muscle fibers attached to the surface of the bone. While this ribbon here is formed of the decalcified compact bone. In the inner side here, you can see the bone marrow. First, the bone marrow is diagnosed by the presence of these empty spaces representing the fat cells, and these dots are representing the hemopoietic tissue. The skeletal muscles are diagnosed by these polygonal cut transverse fibers, and you can diagnose that the nucleus is peripheral in position. And this middle area of the ribbon of the decalcified compact bone, in this ribbon you can diagnose the following. You can diagnose these Haversian blood vessels. Here we have the Haversian blood vessels, this one and this one. All of these are the Haversian blood vessels. if you have a label for these small dots here, these are the osteocytes present in their lacunae or osteocytes in lacunae. So in this area, we don't have except these Haversian blood vessels. If you have a label for this transverse blood vessel, it is an anastomosing vessel between two Haversian blood vessels. After that, we have these dots that will be labeled as osteocytes in lacuna. This is a picture for the decalcified. It is a stained, compact bone. It has Haversian system. In this section, you can see here, this area is the Haversian blood vessel. The Haversian blood vessel, here, this is the Haversian blood vessel, and this is a Haversian blood vessel also, and it is surrounded by concentric bony lamellae. These are the osteocytes in lacuna. You may be asked here about the feature, the LM feature for the osteocyte. It is oval, it is basophilic in staining. Nothing else can seen here in the light microscope picture in this slide. This circle is called Haversian system, and this area is called interstitial lamellae, and this area in between the osteocytes are called concentric bony lamellae. These fine lines are processes arising from the osteocytes so that they can exchange with each other the nutrition, the waste products, as the matrix is calcified and no tissue can pass through it. You may be asked about one or two functions for the osteocytes which are evident in all this picture. The functions, they are bone maintaining cells. Bone maintaining cells responsible for the maintenance of the bone. How? By formation both the organic part of the matrix and calcification of the matrix, which is the inorganic component of the matrix. And this is through deposition of calcium salts, as they have an alkaline phosphatase enzyme. Here, if you are asked about one difference between the osteocyte and the chondrocyte, you can say that the osteocyte, one osteocyte is present in its room, which is the lacuna, not evident here, while the chondrocytes can, as being capable of division, can present in groups. This picture on the right side demonstrates the decalcified compact bone as it is stained, and this label here will be the Haversian blood vessels. Why I say Haversian blood vessels and not Haversian canal? Because the decalcified compact bone section does not contain except the living tissues, does not contain calcified tissue, so you should say Haversian blood vessel. If you have a label for these dots, these are osteocytes in lacuna. If you have a label for this area here in between the circular Haversian systems or osteon, this area in between is called interstitial lamellae. So if we have a label for or a circle surrounding this area, it is the Haversian system or osteon. If you have a label in the area in between the Haversian systems, here or there or there, these are called interstitial lamellae. If you have a label for this blood vessel, it's better not to mention Volkmann canal because it is the anastomosing vessel passing in the Volkmann canal, as I cannot see the Volkmann canal, which is a calcified tissue, in the decalcified compact bone section. So it's better to say anastomosing vessel passing in between two Haversian blood vessels. It's better than mentioning the name of Volkmann canal, or it's more correct. In the second picture also, as it is stained, so it is a decalcified compact bone. What we can see here? You can see here in this photo, you can see here the Haversian blood vessel. Haversian blood vessel. What are these dots in circles around the Haversian blood vessels? They are osteocytes in lacunae. So these are the osteocytes in the lacunae, and this circular part is called Haversian system, and these lamellae are called bony lamellae, and as they are present at the position parallel to the outer circumference of the bone, they are called external circumferential lamellae. So these lamellae parallel to the periosteum, here we have the periosteum, these lamellae parallel to the periosteum are called external circumferential lamellae, while these lamellae are the lamellae of the osteon or Haversian system. If we have a label for lamellae in between Haversian systems, it is called the interstitial lamellae. So in this picture we have the periosteum, we have the external circumferential lamellae, we have the Haversian system or the concentric lamellae, and we have interstitial lamellae, and we have the Haversian blood vessel. This is a very nice picture. You here can see the Haversian system here. You have a Haversian system here. This Haversian system is formed of Haversian blood vessel surrounded by concentric bony lamellae, and here we have the interstitial lamellae. Here we have the concentric bony lamellae, and the blue dots here are the osteocytes in their lacunae, or osteocytes alone, because lacunae cannot be seen in this section, which is decalcified. Here we have the blood vessels also here, also blood vessels here and there and there. All these are blood vessels, and you can see that they are anastomosing. We have an anastomosing vessel arising from this one to join a neighboring Haversian blood vessel. So this is also the ground compact bone, but you can be asked about the functions of the osteocytes, the LM picture of the osteocyte, the enzyme present in the osteocyte. This is for this picture. This is the picture for decalcified compact bone, which is this layer, and inside the cavity of this decalcified compact bone we have the bone marrow characterized by these multiple fat cells. And this is the ribbon of the decalcified bone covered by transverse section of the skeletal muscle. Here we have the internal circumferential lamellae. Here we have external circumferential lamellae. Here we have the Haversian system. In the center we have Haversian blood vessel, and surrounded by this concentric layer of osteocytes in their lacunae. So these big spaces are the Haversian blood vessels. All of these. These small dots are the osteocytes inside their lacunae. And this is an anastomosing vessel between the two Haversian blood vessels. This is very clear Haversian system or osteon. And in between we have interstitial lamellae. This is a picture for decalcified compact bone. Why it is decalcified? Because it is stained. Why it is compact bone? Because I can see the Haversian systems. This is the Haversian blood vessel inside the Haversian canal. And these are the concentric lamellae of the osteocytes in their lacunae. And these are the interstitial lamellae here, and this is an osteocyte in its lacuna, and these are Haversian blood vessels inside Haversian canal. This is also a decalcified compact bone, and it shows Haversian blood vessel inside the Haversian canal, and this is the endothelial lining of this blood vessel. These are the concentric layers of the osteocytes inside their lacunae, and in between we have the lamellae formed of calcified collagen fibers. Here we have, this is the lacuna, and this is the osteocyte inside the lacuna, and here we have the collagen fibers calcified, and this is also the space where the osteocyte is present in it. Also decalcified compact bone, Haversian blood vessel lined by endothelium, and this is the blood, and this is present in the Haversian canal. And all of these are osteocytes in their lacunae, and these areas are the calcified collagen fibers, red in stain. And this is an anastomosing blood vessel which should pass through the Volkmann's canal. Another nice picture for the ground compact bone. This arrow here is for the Haversian canal. This is arrow for Haversian canal. And these arrows here, they are denoting the concentric lamellae. These are concentric bony lamellae, and these concentric bony lamellae actually they are showing canaliculi. So it's better to mention the two words because you don't know which one is needed by the examiner. I can see here canaliculi, and there, and there, and there, and at the same time these are concentric lamellae, so they are concentric bone lamellae showing canaliculi. But this arrow here is denoting the lacuna, and here these two arrows here are denoting the canaliculi. Here are the canaliculi radiating from the lacuna. So this area in between, you can say it is the concentric lamellae or the canaliculi, so it's better to say concentric lamellae showing canaliculi. Now this is the other way of preparation of slides for the compact bone, which is the ground compact bone. In this slide you cannot see cells, you only see the calcified tissue. First of all, this is the calcified tissue, which is the Haversian canal, and it is black due to the entry of air after the death of the cells, as we don't produce or don't perform fixation in these slides. Then we have here the canaliculi connecting the lacunae together. So these oval spaces, black in color, are the lacunae, or these are lacunae. But these connecting channels here, they are the canaliculi. This is the lacuna, and these are the canaliculi. And of course, this is the Haversian system in the ground form of preparation of bone. This picture is unstained, and it is very light yellow in color. So this is the section of the ground compact bone. You should mention 2 points in this identification: the method of preparation, which is grinding method, and this method does not involve any staining, any fixation, any staining. So by this method you can only see the calcified tissue in the bone, and you never see living cells. So this is the ground, and it is compact because it has the regular arrangement of this Haversian system here, so it is a compact bone and not a spongy bone. Moreover, the spongy bone cannot be prepared by the grinding method as it is very fragile, so it will be damaged by this method. In this picture, you may have a label for this white or black circles, these are the Haversian canals and not Haversian blood vessels. It is a calcified tissue, not a living tissue. In the ground compact bone, we have the calcified tissue, so it is the canal which normally contains inside the Haversian blood vessel. But the Haversian blood vessel cannot be seen in this picture. We only see the room which is the Haversian canal. Each Haversian canal is surrounded by concentric lamellae of the calcified lamellae or calcified collagen, and we have in between these calcified lamellae we have the rooms of the osteocytes. So if we have a label here for this area, these are the concentric lamellae, and if we have a marked enlarged picture and we have a label for this black dot here or black line, it is the lacuna. And if we have a label for this area here, this area of that is present in between the Haversian system, this is the interstitial lamellae. These are the circumferential lamellae here, circumferential lamellae, and this is the Haversian system, and this is the Haversian canal. And if we have a label for this black line, it is the lacuna of the bone. Yeah, it's better not to mention the word osteocyte. It is the lacuna. So this is the picture of the ground compact bone. Another picture for the ground compact bone. Of course, this black space is the Haversian canal, and this Haversian canal, and that Haversian canal. These are the concentric bony lamellae showing canaliculi, and this is the interstitial lamellae in between the Haversian systems. Another picture for the ground compact bone, we have here the Haversian. Here we have the Haversian canal, and surrounded by the concentric bony lamellae here, and these lamellae are alternating with the lacuna here, and from the lacuna we can see very clearly the canaliculi. Here we have here the canaliculi radiating from these lacunae. These minute threads are the canaliculi radiating from the lacunae. Actually, in a section of the bone, you may be asked about the function of these canaliculi. What is the function of these canaliculi? They are the passages through which processes of osteocyte can reach the processes of the neighboring osteocytes, so that the nourishment can pass from one cell to another very easily. And this is also a picture for the ground compact bone, and these are the Haversian system formed of the concentric lamellae. That's to say they have a common center. And these lamellae are formed of these lacuna containing the osteocytes, of course, and in between we have the calcified collagen fibers. This is the Haversian canal, Haversian canal, Haversian canal, Haversian canal, and these areas black here, they are the lacunae, and the fine threads are the canaliculi. We don't have any living tissue in this slide. Here also we have the ground compact bone. This is the lacuna, and these are the canaliculi. This is the Haversian blood vessel in the Haversian canal, and this is an anastomosing blood vessel from one canal to another. It is present in what is called Volkmann's canal. So this is the Volkmann's canal, and this is the Haversian canal. Because we don't mention any living tissue in this slide. We have Haversian canal, calcified tissue. We have Volkmann's canal, calcified tissue also. And these are the lacuna, and these are the canaliculi. also Haversian canal, and here all of this structure will be the Haversian system. This is a picture showing the spongy bone. When you look at this picture, you can diagnose that we don't have here a regular bone marrow cavity. We have irregular bone marrow cavities here, and we have bony trabeculae, which are also irregular. No Haversian system can be seen here. So this is the spongy bone. You may be asked about the size of the spongy bone. Of course, you all know that the small or irregular bones or the epiphyses of the long bones are all made of spongy bone. In this picture, you should diagnose the following. You should diagnose these cells on the surface of these bony trabeculae. These cells on the surface, they are free cells, so these are the osteoblast. You may be asked for one or two features that can be seen by the light microscope for the osteoblast. Of course, you can diagnose they are oval in shape and they are basophilic. Here it is darkly stained or basophilic in staining. And mention one enzyme present in them. It is the alkaline phosphatase enzyme. Mention two functions. They are the bone forming cell. They secrete both the organic part of the matrix and the inorganic part of the matrix, which is the calcium. They are responsible for the deposition of this calcium in the matrix due to their content of alkaline phosphatase enzyme. And once they are imprisoned in rooms called lacuna in the bony trabeculae, they are called now osteocytes. So these are the osteocytes. Inside the bony trabeculae, they are osteocytes. On the surface, free cells, osteoblast. One difference between this and that, it is more basophilic, less basophilic. This is not surrounded by matrix, this is surrounded by a matrix. And this red area is the bony matrix here, which is formed, of course, of the collagen of type 1. After that, we have the irregular bony cavities, which is full of the different cells responsible for hemopoiesis. This is the bone marrow cavity, and you may be asked about what the type of this bone marrow. It is a red because it is present in the spongy bone, so it is a red or active bone marrow. So we have here osteocyte, osteoblast, bony trabeculae or bony matrix, bone marrow. Another nice picture for 3 osteoclasts. Here, multiple nuclei, acidophilic cytoplasm, and they are present on the surface of the bone, lining the bone marrow cavity. Inside the bony trabeculae, we have the osteocytes, and here we have a blood vessel in the bone marrow cavity and hemopoietic cells in the bone marrow cavity. Nice picture for the spongy bone. Here you can see the irregular bony trabeculae, which is acidophilic in staining. And in the picture also you can see these irregular bone marrow cavities. So you will describe or you will diagnose structures in the bony trabeculae and in the bone marrow. In the bony trabeculae, on the surface, as you see, we have dark oval cells, free, not imprisoned in matrix, so they are called osteoblast. And inside the bony trabeculae itself, we have imprisoned cells, they are the osteocytes, which are responsible for the maintenance of the bone, and this is for building of the bone. Also, in the bone marrow cavity, you can see these blood vessels and these hemopoietic cells responsible for the production of the different blood cells. Nothing else here in this picture. This is a very nice picture for the spongy bone. Here we have in the spongy bone, we have the bony trabeculae. First of all, this is the picture with the original size. These are the bony trabeculae here, very evident. And in between we have irregular bone marrow cavities. In the bone marrow cavity, I can diagnose this large cell with single multi-lobed nucleus. It is the megakaryocyte. And we have the different hemopoietic cells. In the bone marrow trabeculae, in the bone spongy bone trabeculae, I can enlarge the picture more and you can diagnose what? Here in the bony trabeculae, here this is the red bony trabeculae, we have the osteocytes inside. While in the lining here we have a lining cells here, these cells here are the osteoblast. So the osteoblast on the surface of the bony trabeculae and the osteocyte inside the bony trabeculae itself. Now we have here also the megakaryocyte. You can see the megakaryocyte here with a single multi-lobed nucleus. This is another picture also demonstrating the osteoclast. Acidophilic cytoplasm due to the presence of a huge amount of lysosomes, membranous organelle, and we have multiple nuclei, and they are related to the surface of the bone in the bone marrow cavity. Here we have the osteocyte inside the bony trabeculae, and we have here the bone marrow cells. This is another picture also for the spongy bone. We have here the bone marrow cavity, here in the bone marrow cavity here. All of this is the bone marrow cavity. It is lined by the endosteum, which contains the osteoblast. Here we have the bony trabeculae, which is formed of the acidophilic bone matrix, and we have these imprisoned cells, which are the osteocytes. So this is the endosteum, very thin layer, which is rich in osteogenic cells and osteoblast. Here we have the bony trabeculae containing the matrix as well as the imprisoned osteocytes. Another two nice pictures for the spongy bone trabeculae. We have here the spongy bone trabeculae or trabeculum, because it is a singular, and inside it we have the osteocyte, and on the surface you can see here two types of cells lining the bone marrow cavity. We have here the oval basophilic cells with single nucleus, which are the osteoblasts. This is the bony trabeculae. This is the bony trabeculae. These are with the star is the osteocyte. But this cell, which is very large cell, and it is multinucleated, and it is not so dark like this one, as it has an acidophilic cytoplasm. This cell is the osteoclast. This is the osteoclast, which is the bone destroyer cell or the bone eating cell. In this picture, you may have questions about the functions of the osteoblast. We have said them. Functions of the osteocyte, we have said them, and also the functions of the osteoclast. The osteoclasts are responsible for bone resorption by secretion of carbonic anhydrase enzyme, and this carbonic anhydrase will form carbonic acid that will dissociate to give hydrogen ions. The hydrogen ions will dissolve the calcium. Not only that, but they secrete also lysosomal enzymes which will cause the lysis of the organic part of the matrix. So these cells will cause the removal of both the organic and the inorganic. The inorganic by the release of hydrogen ions, causing lysis or causing the dissolving of the calcium, and the organic material is also dissolved or hydrolyzed by the lysosomal enzymes. Also, they have a very important role in bone remodeling during the ossification. In between these bony trabeculae, you can see these are the bone marrow cells and blood vessels in this bone marrow. Here in the other picture also, you will see the bony trabeculae and the osteoblast and the osteocytes here in the bony trabeculae. So we have the osteoblast, which are dark and with one nucleus, and the osteocytes in the bony trabeculae. Here we have also a covering on the surface of this spongy bone. It is formed of the periosteum. So in this picture we have the periosteum, we have the bony trabeculae, we have the osteocytes, we have the osteoblast bone marrow cavity. This picture is a very nice picture. We have here 4 pictures. We will start by this one on the right side. Now, what we have in this picture? Actually, these are the osteoclasts. The osteoclasts are multinucleated. These are the nuclei present in the osteoclast. We have here 4 osteoclasts. Then we have this area which is called brush border, like a brush, and it is towards the surface of the bone. This is the bone, and this is towards the surface of the bone. At this brush border in the osteoclast, the brush border is the area in which there is a microenvironment which is of low pH, and lysosomal enzymes are released here so that bone resorption takes place. Accordingly, we have an empty space. Look at this small arrowhead. This empty space due to resorption of the bone is called Howe-Ship's lacuna. It is called here Howe-Ship's lacuna. So, this picture demonstrating these 4 osteoclasts. This is the bony surface, and we have here the multiple nuclei, and this cell is responsible for bone resorption and of course for bone remodeling during ossification. After that, we have another picture. We'll see it here on the left side. On the left side, we have also the osteoclast. Here we have the osteoclast. The cell is an osteoclast, characterized by being multinucleated, acidophilic cytoplasm, and the space around it is called Howe-Ship's lacuna. And the cause of this space is the release of the hydrogen ions and lysosomal enzymes. The hydrogen ions causes the dissolving of the calcium present in the bone, and the lysosomal enzymes cause the lysis of the organic part of the matrix. And these are bony here trabeculae. Another picture also we have here, this one. This one is also for a very nice osteoclast, as we see here, looking like a turtle. It has multiple nuclei, it has an acidophilic cytoplasm, and it is surrounded by this space, which is now very, very wide space, Howe-Ship's lacuna. And here we have the bony trabeculae. Again, we have another one also for the osteoclast. these are three osteoclasts. We have this bony surface where bone resorption takes place. So we have here, this is the bone, and we have also inside the bone itself, here we have this osteocyte inside the bone, but these are the most important feature in the photo, these osteoclasts, and this is the bony trabeculae. And we have here other cells also on the surface of the bone. We have osteoblast. Inside the bone we have osteocyte. This is a picture for the spongy bone, where you can see at the first look the multiple irregular bone marrow cavities, characterized by the presence of many fat cells that appear as white empty spaces. And in between these cavities we have the spicules of the spongy bone. And by higher magnification you can diagnose the osteocytes in these spicules, while the osteoblasts are present lining the bone marrow cavity. You should know what are the sites of this spongy bone. This is also a picture for the spongy bone, and you can see here the multiple bone marrow cavities here, and you can diagnose also a very nice cell here inside the bone marrow, which is the megakaryocytes formed of this multi-lobed nucleus and within this big size. Here we have also the osteocytes inside the bony trabeculae, and these bony trabeculae which are facing the bone marrow. You can see here in the lining of the bone marrow the osteoblasts. So all of these are bone marrow cavities, and these are the spicules of the bone. Of course, all of these are the blood cells. The bone cells are very important subject for questions in the data show. You should know that we have the osteogenic cell, the osteoblast or the bone building cell, the osteocyte, which is the mature bone cell, and the osteoclast, which is the bone destroying cell or bone resorption cell. In each cell you should study by heart the origin, the site, LM, EM, and functions. And it's better to make a table for them for easy recalling during the exam. Here we will start by the osteogenic cell. As regards the osteogenic cell, origin from the UMC. As regards the light microscopic picture, or the site first, the site, it is present in the inner layer of the periosteum and endosteum. يبقى هنا هتكون موجودة في ال covering and ال lining of the bone, and in the inner layer and not the outer layer, because this periosteum has an outer and inner, so it is present in the inner layer of the periosteum as well as the endosteum. Then we have the light microscopic picture. As regards the light microscopic picture, they have the following features. As the shape, first of all, they are flat cell, and they have flat nuclei, and they have a pale basophilic cytoplasm. Then, what is the EM picture for this cell? The EM picture here, it shows the signs of immature cell in the form of just free ribosomes to produce the proteins of the cytoplasm of the cell, just free ribosomes. As regards the functions here, actually they are responsible to give rise to the osteoblast, the bone building cell, and also they can give rise in case of the poor vasculature. This osteogenic cell can give rise to the chondroblast in area of poor vasculature of the bone. Then We will talk about the other cell, which is the osteoblast or the bone-forming cell. As regards the origin, we have said it is from osteogenic cell. As regards the site, it is present immediately under the periosteum. So it is very near to her mother, periosteum. As well as the endosteum, so it is very near to her mother, which is the osteogenic cell. So in the periosteum, under the periosteum and the endosteum. Then we have the LM. As regards the LM, it is oval cell with minute processes. And the cytoplasm is darkly basophilic, showing a negative Golgi image. So we have here a negative Golgi image like that we see in the plasma cell. This means a well-developed Golgi, and this means also that the cell here is forming proteins in large amount. So here we have an oval with few processes. This is the shape. As regards the cytoplasm, it is deeply basophilic and showing negative Golgi image. As regards the nucleus, it is an oval nucleus with the eccentric. It is eccentric in its position, and it is achromatic, and it is pale with prominent nucleolus. Again, what are the L microscopic pictures or features of this osteoblast? It is an oval cell with few processes. This is the shape. Then we have the nucleus. It is an eccentric in position, and it is pale with prominent nucleolus. Then we have the cytoplasm, deeply basophilic with an evident negative Golgi image. Now let's move to the EM. What you will expect? I will expect that they have a well-developed factory for production of the protein. They have well-developed rough ER, Golgi, mitochondria, in addition to the achromatic nucleus. Then lastly, what about the functions of these cells? These cells secrete the alkaline phosphatase, and which will form the calcification of the bone. And also it secretes organic material of the matrix. And don't forget that the newly forming organic material is called osteoid. Osteoid. These cells, after being surrounded by the matrix they secrete, they are transformed now to osteocytes. So here we can mention that the functions will be the following. Number one, here secretion of the inorganic material, which is the calcium salts, by the aid of the alkaline phosphatase. Number two, formation of the organic material, and when it is newly formed it is called osteoid. Oid means shabah because it is still immature. Osteoid. Number three, it is when it is imprisoned inside the matrix, they are now called osteocytes. And finally, we can say that it is a bone-forming cell. Then we have the osteocyte, which is the mature cell here. We will move these features and functions for the osteoblast to talk about the osteocytes. As regards the osteoblast, they are the mature bone cells. What is the origin here? Origin is from osteoblast. What about the site? The site, they are present inside lacunae, inside the structure of the bone. They are present in lacunae, and these lacunae are connected together by canaliculi. So this is the cell that I will draw now. It is the cell that have processes, very fine processes that passes through these canaliculi to reach the adjacent processes from the neighboring cell, and in between here we have gap junction, so that any nourishment here will be easily passing to the neighboring cell. And that's why we say that the cells in the bone are well nourished. Then we have to talk about the LM. The LM is that the cells here are oval, as you see, and it have minute fine processes, and it has an oval nucleus. The cytoplasm here is the reverse of the osteoblast. It is less basophilic, and the nucleus is more dark. Why? Because the activity here is less than that of the osteoblast, and accordingly we can expect that they have less mitochondria, less Golgi, less RAF ER, and they have an heterochromatic nucleus because the nucleus here is much darker than that of the osteoblast. Don't forget also in this point to mention the gap junctions between the processes of the neighboring osteocytes. As regards what the functions, they have the power to form the organic matrix exactly like the osteoblast, and the calcification also due to the presence of the alkaline phosphatase, and they are important for the viability of the matrix. It is responsible for maintenance of the bone structure or bone matrix by formation of organic material and inorganic material, and by that they can make the bone viable and healthy. Don't forget also that they cannot divide. Now let's talk about the last cell, which is the osteoclast. Here, this is the osteoclast. Here, what about its site? It is present inside the bone marrow in a cavity called Howe-Schepps lacuna. So the site here, it is present in the Howe-Schepps. This is Howe-Schepps, and you will add apostrophe S lacuna. Type. What about the origin? It is originating from the blood monocytes. They are originating from the blood monocytes, which are characterized by having kidney shape. By these cells, or from this cell, fusion of multiple monocytes will give rise to this osteoclast, so that we can expect that it has multiple nuclei. As regards the LM picture, here the LM picture, LM. Here the shape. First, it is a large irregular cell having a border looking like a brush زي فرشة الشعر. This is the shape. As regards the nuclei, they have multiple nuclei. Not a single nucleus like that of the megakaryocyte, which is also present in the bone marrow. No, it has multiple nuclei as it originates from the fusion of the blood monocyte. Also very important to know that these nuclei are from 6 to 12. And also don't forget that they have a foamy, زي رغاوي, foamy appearance for the cytoplasm, which is acidophilic. Then as regards the EM picture, here you can mention that it has a border which is ruffled, called ruffled. This this irregular border, which is called brush border in the light microscope, it is called a ruffled surface, which shows the microvilli. Here, this surface has microvilli. And the cytoplasm here this is cytoplasm, shows what? Shows the multiple lysosomes as they secrete these lysosomal enzymes to cause resorption of the bone. In addition to, of course, to the Golgi apparatus and mitochondria, but very important feature is the presence of the ruffled border in the form which are microvilli, صوابع صغيرة, and cytoplasm is rich in the lysosomes. Then, as regards the functions, here, don't forget that it is a bone resorbing or bone destroying cell. So its function is the resorption of the bone. And this takes place in 2 steps. Step 1, it contains an enzyme called carbonic anhydrase enzyme. This carbonic anhydrase induce this reaction. Water with carbon dioxide will form the carbonic acid. And this carbonic acid will dissociate to bicarbonate plus the hydrogen. And this hydrogen will be released from the ruffled border, causing acidification of the matrix that will dissolve the calcium salts. يبقى هنا لما بيطلع الهيدروجين ايونز دي في الماتريكس هتعمل dissolving للكالسيوم salt. So at first we have to remember how it dissolves the inorganic material. Then the second step to get rid of the organic material. And this takes place by the release of the lysosomal enzymes, which will cause the lysis of the organic material. Lysis of the organic. So it has 2 jobs, is removal or the dissolving of the calcium salts by the secretion of carbonic anhydrase, and the lysis of the organic material by the release of the lysosomal proteolytic enzymes present inside the cytoplasm, inside the lysosomes. Now look to the picture again here. This is the picture we have, and we'll summarize what we have said. Here, this is the osteogenic cell, the osteoblast, the osteocyte, and the osteoclast. In this picture, this is the osteogenic or the osteoprogenitor cell. And these are the daughters of this cell, which are called the osteoblast. And the newly formed matrix is called osteoid. And this is a mature bone cell with the processes passing through the canaliculi and forming a gap junction with the neighboring processes. As you see here, this is a process and this is a process, and in between we have gap junctions. And this is the bone matrix, and this is the osteoclast, the bone resorbing cell, and this is the house-shaped lacuna due to the dissolving of the organic as well as the inorganic material. This is a picture for the spongy bone, and as you see here, this is the bony trabeculum. It is formed of this matrix, acidophilic, and it shows the lacuna with the osteocytes inside. So these are osteocytes inside lacuna. And in between these trabeculae, you can see the bone marrow cavities, and here we can see a blood vessel to take the newly formed blood cells, and you can see multiple fat cells. But here, lining this cavity, you can see other cells, which are the osteoblast on the surface of this trabeculum. And they are having one nucleus, so these are the osteoblast and not osteoclast. Here also osteocyte within the lacuna, and this another bone marrow species. This is more magnified picture for the spongy bone, and also you can see the matrix acidophilic. You can see the osteocytes inside the lacunae. You can see on the surface of this trabeculum the osteoblast. And this, of course, demonstrates how the spongy bone is formed by intramembranous ossification. It is an intramembranous bone formed by intramembranous ossification. This is a blood vessel showing the RBCs and nothing else here. And also here RBCs in a blood vessel. Another picture for the spongy bone. Here this is the trabeculum of the spongy bone, and here on the surface these cells which are having one nucleus, it is the osteoblast. Inside it is the osteocyte in lacuna, and this big cell which is formed of many nuclei, and it is present also on the surface or in the bone marrow cavity very near to the bone surface, as it is a bone resorbing cell. It is the osteoclast. How did you know? From the multiple nuclei, from its site on the surface of the bone, and from the big size acidophilic cytoplasm. It is a picture showing the stages of ossification in the epiphyseal plate. At first, here we have the head of the bone formed of spongy bone. And of course, it is covered by compact bone, which is not evident in the picture. Then we have, this is the epiphyseal plate. At first, we have an area of normal hyaline cartilage, and here the cells are discrete from each other, and this area is called resting zone. Then the cells begin to increase in number by mitosis. So we have columns of cells above each other. So here we have the discrete cartilage cell, and then we have the cells have proliferated in number, increase in number, and became arranged above each other like the coins. Then, after that, we will have this increasing in the size. This increase in size is called hypertrophy. And the increase in size, you may be asked the cause of this increase. The cause will be the accumulation of the glycogen and alkaline phosphatase enzyme. After that, these cells will die and leave empty spaces. And the cause of the death is the deposition or the calcification that takes place in the matrix and in the periosteum, so that no nourishment takes place and the cells will die. This death will allow the invasion of the undifferentiated mesenchymal cells and osteoblast to reach this area and form the irregular bony trabeculae of spongy bone. After some time, these irregularities will be removed and the osteoblast will form concentric lamellae of compact bone, and complete ossification and complete compact bone formation will take place. So this area is the resting, this is the proliferating, this is the hypertrophied, and this is area in which we have spicules or trabeculae of spongy bone is called stage of spongy bone formation or ossification. This is a picture in the growing end of the bone at the region of the metaphysis, which is present between the epiphysis and diaphysis. And it demonstrates the intracartilaginous ossification stages. First, here we have the resting cartilage cells, and then the proliferation where there is an increase in the number. Then we have the increase in the size, which is the hypertrophy. Then we have the zone of cell death, where the cells, due to the calcification of the matrix, will die, and here you can see the nuclei are in a condition of disappearance. And then this will be followed by zone of invasion. Here the empty areas resulting from the death of the cartilage cell will be invaded by the blood vessels and undifferentiated mesenchymal cells. Then these undifferentiated mesenchymal cells will form osteogenic cells that will form spicules of the bone. At first, the bone is in the form of a spongy bone, and then complete ossification, where the spongy is changed to a compact bone, takes place. Name the stages marked by the red arrow. It is the stage of proliferation, where there is an increase in the number and they look like coins over each other. And the yellow circle here, it is in the stage of hypertrophy, where the cells are bigger in size. And then the green circle, it is the stage of calcification, where the calcification takes place in the matrix, leading to the death of the cells, as you see here. Identify the tissue. It is the decalcified compact bone. Name the structures pointed out by the yellow arrow. Here it is the Haversian blood vessel inside Haversian canal. Blue arrow, it is the osteocyte in the lacuna. And number 3, the yellow outline constitutes here the interstitial lamellae, which are the lamellae present in between the Haversian systems. The star represents the bony matrix. Its acidophilia is due to collagen fibers. Mention the type of junction connecting similar cells pointed out by the green arrow. These are osteocytes, so the junction between the cells is the gap junction. And what is the value of that? It is the easy transport of nutrients from one cell to another. Then we have name the cell pointed by the blue arrow and the enzyme it contains. The blue arrow is denoting the osteoblast, and it contains the alkaline phosphatase. Here we have a trial exam. Here we are asked to identify the tissue. It is compact bone, and the method of preparation, the method is by grinding, and it is called ground compact bone. Identify one is the Volkmann canal, 2 is the Haversian canal, 3 we have the lacunae, 4 we have Haversian system or osteon, 5 we have concentric lamellae containing canaliculi, 6 here it is also denoting the canaliculi, so you can mention in 5 the concentric lamellae, and in 6 it is the canaliculi. Identify A and B. A is the Haversian system. B is the Haversian blood vessel inside the Haversian canal. And the asterisk is pointing to the interstitial lamellae. The arrowhead points to the osteocytes inside their lacunae. Name the cells A and B. A, of course, it is the osteoclast. How did you know? Because it is multinucleated, having an acidophilic cytoplasm on the surface of the bony trabeculum. B here, it is also on the surface of the trabeculum, but it is oval cell with one nucleus and a basophilic cytoplasm. It is the osteoblast. The yellow outline marks the Haversian system. The red arrow points to Haversian blood vessel inside Haversian canal. The cell pointed out by the blue arrow is here it is the osteocyte. And mention its main organelles. This cell is synthesizing protein, so the main organelles will be the rough endoplasmic reticulum and the Golgi apparatus together with the mitochondria. The yellow arrow points to the osteoclast. Its origin is from monocyte. The shape of cells pointed out by the green arrow, it is the shape denotes it is an active cell, indicates it is an active cell. Actually, it is the osteoblast. Name the structures A and B. Here A, it is passing transversely from this canal to other, and these canals are parallel to each other. This is transversely passing, it is the Volkmann canals. What is the function? If you are asked, to carry an anastomosing vessel between two Haversian blood vessels. And the B here, it is the passing parallel to the longitudinal axis, it is the Haversian canal. What is the function of this canal? It carries the Haversian blood vessel for the nourishment of the bone. If you are asked what is the type of this slide, it is a compact ground compact bone, because we don't see here any living cells, we are just seeing the calcified tissue. And we don't have a ground slide for the spongy bone, as it is very fragile and friable and will not be available to us to produce a ground section. This picture is a very good exercise for the ground compact bone. What is the blue arrow pointing to? This is the blue arrow, so this is the Haversian canal, Haversian canal, Haversian canal, Haversian canal. What is the area that the white dotted lines are outlining? This area is the interstitial lamellae. They are the lamellae present in between the osteon or the Haversian systems. What are the yellow dotted lines showing? These dotted lines are showing the concentric bone lamellae. Concentric because they have one center and they are sharing this one center. These circles are all sharing one center, so they are called concentric lamellae. What is the area dotted by the green dots? It is the osteon or Haversian system. What the arrow, white arrows point to? The white arrow here points to the room occupied by the osteocytes. So this room is called lacuna. So this is lacuna, this is the Haversian canal, concentric lamellae, Haversian system or osteon, interstitial lamellae. This is a picture for the decalcified compact bone. Identify the tissue, it is compact bone. The method is the decalcification. If you are asked about how decalcification takes place, it is by putting the bone in nitric acid 10% to dissolve the calcium, and after that the tissue becomes soft and it is fixed and stained. Identify 1 and 2. Here 1 is the Haversian blood vessel and 2 these are the osteocytes. Identify the structure labeled by the red. The red here arrow denotes the osteoblast, which are present on the surface of the red bony trabeculae, and it is free in the bone marrow. These are the lining or present in the endosteum, outlining the bone marrow cavity. So these are the osteoblast. Green here the imprisoned cells called osteocytes. Blue arrow is the bone marrow cavity, and of course, as it is in the spongy bone, it is a red bone marrow. Blue arrow and red and blue star. Red star is the bony trabeculae, and it is of course here present in between the bone marrow cavities, and it is acidophilic, containing osteocytes inside. This is a very nice picture demonstrating the stages of ossification. Here, first of all, we have the stage of resting cartilage, where the cells are discrete. Here the stage of proliferation where the cells increase in number and became arranged above each other like the columns of coins. Here in the stage of the hypertrophy where the cells are large in size, and this is due to the accumulation of glycogen and alkaline phosphatase. And this is the stage where the cells die and the area is invaded by the undifferentiated mesenchymal cells and osteoblasts to form irregular acidophilic bony trabeculae. In between we have bone marrow. This is the stage of ossification and formation of the spongy bone. Another picture here. What are the green arrows pointing to? The green arrows here evidently are pointing to these large cells, acidophilic, multinucleated, on the surface of the bone, very near to the bone marrow cavity, and these are called the osteoclast. What is the name of the space where the yellow dotted line? Yes, this is space, which is the clear space on the surface of the bone related to the osteoclast. It is the Howe-shaped lichen, and you may be asked about how this space is formed, and I have answered that before. What are the cell types that the yellow arrow is pointing to? Here they are evident. This yellow arrow is pointing to this osteocyte present in the bony trabeculae. So we have an osteocyte present in the bony trabeculae. We have osteoblast on the surface. We have also on the surface a large cell. This cell is different from that in two forms. This is small, this is large, this is single nucleus, this is multiple nuclei, and this is basophilic, and this is acidophilic. This is another slide for exam. What is the name of the region outlined by the green arrow? This region, all of this region with the multiple fat cells, with the dots demonstrating hemopoietic tissue, with the large cells in between, you can see these are the megakaryocytes. So this is the bone marrow. Red arrows point to. The arrows here points to acidophilic, the bony trabeculae. Here we have the compact bony trabeculae here. And the yellow circles enclose, here enclose the Haversian system. If you are asked about the type of the bone marrow, as this bone marrow is present inside the compact bone, you can say that it is a yellow bone marrow as it is present in the cavity of a compact bone.